In this video, we'll be going over transformation. Before we do that, we need to understand all the previous other steps. So we're doing transformation today. We need to understand all of these three steps. Isolation. So we should know that we are isolating our desired DNA fragment, our desired gene, maybe an insulin gene. And there are different ways of isolating that. And then number two is using restriction endonucleases. Very important enzyme to have those sticky ends. So you can have a sticky end in your desired gene, and using the same restriction endonucleases on the plasmid, cut the plasmid out, having the same complementary sticky ends. And now we have two things: plasmid with sticky ends and our desired DNA with sticky ends. Then we ligate them together. We join them together using ligase, and that's our ligation or insertion. And then what we do, we transfer. So now that we have a plasmid, that plasmid is a bit different now. Why? It has your desired gene inside it. So what do we call that? Recombinant plasmid. Now that recombinant plasmid will be transferred into a host cell, into a bacterial cell, and that's called transformation. And that's our lesson today. So now there is two scenarios that we can have. In here I'm showing you my recombinant plasmid. Why is it recombinant? It's plasmid. It's recombinant because it's got this it's got this target gene. So this target gene, the one in blue, is not part of the bacteria. Let's assume that it is foreign. So it's recombinant plasmid. So this is our foreign target gene. This is our desired gene. So this whole plasmid here, this is recombinant because it's got this extra gene from another organism, maybe a human insulin gene. Anyway, we want this recombinant plasmid. What do we want? We want this to go inside a bacterial cell. Now, there are two things that happen. Let me move this. There are two things that can happen. Number one is this one here. This is what we want. The bacterial cell takes up the plasmid. It takes up the recombinant plasmid. Very good. And the other scenario is, for some reason, it does not go. So which one do we want? We want this one. This one is not good. Let's assume I have 100 recombinant plasmid. Only, out of that 100, only one of them, 1%, will be like this. So 99% is like this, meaning the bacterial cell does not take up our recombinant plasmid. That's a big problem. So what scientists do, they need to fix it up. So I'm showing you again, scientists need to find a solution to this, right? So I'm showing you another diagram for you to understand. This is my recombinant plasmid. Why is it recombinant plasmid? Because that green or that blue stuff there, this one there, that is my desired gene. That is my gene from a different organism. And then I have my plasmid, and it says ampicillin resistance gene. We know that plasmid carries antibiotic resistance genes. This one is ampicillin. Now we are going to mix. We're going to mix this recombinant plasmid with lots of bacterial cell. This mixing is done randomly. By the way, you can't visualize, you can't see with your eyes bacteria. You can't see with your eyes recombinant plasmid. Everything's happening in a way they can't see. So there's a random collision, random mixing. And out of that random mixing, I have scenario A, scenario B, scenario C. Which scenario do we want to happen? Have a think. Yes. So if you had said yes, that's correct. We want scenario A. We want this host cell, this bacterial cell, to take up the recombinant plasmid. And that is called transformation. This bacterial cell is now transformed. We don't want scenario B and scenario C. Scenario B, what's happening? The recombinant plasmid, for some reason, is not entering the bacterial cell. And scenario B is showing me that a plasmid has entered, but it is not recombinant. It does not contain the desired gene. Is that any good to us? No. Out of these three things, A is the one that we want. Only 1% is A. Out of 100%, only 1% is A. I say that's a very, very small number. So what can we do to increase that percentage? So what scientists do is it's called transformation, right? So when you're trying to reintroduce that recombinant plasmid into host cell, it's called transformation. We add in calcium ions, and number two, we add in, we increase the temperature. It's called heat shock, and these two things makes the bacterial cell membrane pores slightly larger, and that allows that sort of recombinant plasmid to enter more into the host cell. Now, this is what we do. So there are few reasons why this happens. So there are a few reasons why only 1% enters, right? The number one is some plasmid will have closed up again. So the plasmid that was cut by which enzyme? Restriction endonucleases. It can sort of close back on itself. So that is not good. So you have your plasmid again, and it's not recombinant. And the other thing is the DNA fragment, they can basically join together as well to form their own loop, own plasmid. So that's the reasons why that we only have like 1% going in. So we add in calcium ions, and we add in heat shock. Heat shock is you increasing the temperature to about 40 degrees, and that increases the chances of it going in. For the exam, you don't need to know about the end temperature. Just know calcium ions or heat shock will increase the rate of transformation. That's that success rate. And the three reasons I'm showing you here, what can happen. So whenever you have your recombinant plasmid and a host cell, you have three outcomes. Number one, plasmid opens up and it closes back again, okay, so without the DNA fragment, so you form your own plasmid. Number two, the plasmid is basically takes up the fragment, so it's good, it's got recombinant, but it does not enter the cell, it does not enter the bacterial cell. Number three, the recombinant plasmid basically enters into the cell which we want. That's the green one, and only 1% happens. And the other scenario that we said, that the DNA and the other DNA, meaning we produce maybe more than one fragment. So different DNA fragments, they can join together to form their own loop.